All right, so to finalize interpretive discussion on Chapter 9 notes, just wanted to emphasize a couple of things on here as you look at and get notes from the last slides for yourself. So this kind of repeats the central dogma concept, going from DNA to RNA to protein, because that's what amino acids being combined in sequence are, making protein. Understanding this transition to changing the code slightly as messenger RNA is formed, and then the codons of 3 amino acids forming the code to determine which amino acid comes in what order. I think you get that from the activity from last week. All right, so again, what is associated with tRNA? Most of you already were able to answer that, or it seemed like it in class. And that is each one transfers what to the ribosome? An amino acid. So transfer RNA has an anticodon, a complementary 3 nucleotide sequence that is the complement to the mRNA codon. And they have a specific binding site to attach that specific amino acid to them and taxi them in to the ribosome. The ribosome, think of in 2 parts, kind of like a hamburger bun. There's a bottom and the top. The top is a little bit bigger. And these 2 subunits are primarily made of something called ribosomal RNA, but there's also some proteins mixed in there too. This is a diagram model of transfer RNA, a couple different ones together here. But the idea is that they're carrying the amino acid, and on the other end is the anticodon, which matches with the mRNA codon. So what 2 languages are getting translated at the ribosome? It's going from genetic information, the language of genetic code, into polypeptide, into protein. So from nucleic acid code to protein code. The order of the codons determines the order of the amino acids in the polypeptide chain. So where does translation happen again? Make sure that you keep it straight. Transcription happens in the nucleus at the gene. Translation happens at the ribosome. And that's going to be out in the cytoplasm. Translation occurs in 3 stages. I mentioned, I think, 2 of them in class, and I think I got sidetracked. But the first is when the ribosome parts come together and interact with the start codon. And so that's initiation. And then as the messenger RNA goes through the ribosome and the polypeptide chain is growing, that's called elongation. And then when they separate and the protein is removed, that's called termination. So you hit the stop codon and the ribosome parts separate, messenger RNA separates, the protein that was formed separates. So your book does talk with some detail about each of these, so we'll just remember a few ideas. So the initiation complex is the small ribosomal unit binding to messenger RNA, and then the large one, the large subunit comes in during initiation as well. Start codon, AUG, my favorite month, and it is my birthday month. So that's initiation. Elongation, the polypeptide chain is being formed as the ribosome moves along the messenger RNA. So methionine is the first amino acid in the chain. Codon is AUG. And then once it reaches the stop codon, synthesis ends. Release factors bind to the ribosome. Enzymes detach the messenger RNA and the polypeptide chain from the ribosome. This is just some step art to try to help you kind of visualize the process. I think the videos are maybe more helpful, but this is a diagram from your book. And this is a diagram similar to the one that you were asked to make some sense of for yourself. So transcription happening in here at DNA genes, copying single-stranded messenger RNAs, which then get edited. Exons continue to exist and the ribosomes move along the messenger RNA, polypeptide chain grows, so the transfer RNAs carrying in the amino acids. Make sure you take some time with that diagram. This is a concept that was new to me when I first started teaching this class, and this idea of polysomes. So a messenger RNA might be worked on by multiple ribosomes. So these are multiple ribosomes, one messenger RNA going through and making copies of the same protein. So the protein can be made multiple times by the same messenger RNA using separate ribosomes to do this. And I never really thought about that, but it would make sense to have a whole bunch of ribosomes making a whole bunch of the same protein if the body is in need of a bunch of it. And that wraps up Chapter 9 notes. If you have questions, feel free to ask. And hopefully that made a little bit of sense and isn't too confusing.